In this chapter, you will learn about the cell structure and its function. Cells are the basic building blocks of living things, just like bricks are the building blocks of walls. In this illustration, you will see an animal cell, a plant cell, and a bacteria. These cells vary in size. Cells like animal cells and plant cells are much bigger than the bacterial cells. Please pay attention to this scale bar here. This little black line represents 50 micrometer. This represents 10 micrometer, and that is 500 nanometer, which is half a micrometer. Plant and animal cells have a structure inside the cell called nucleus that houses genetic information. DNA and chromosomes are inside the nucleus. These cells also have internal membrane-bound compartments. Such cells are called eukaryotic cells, whereas bacteria, they do not have internal membrane-bound structure, they do not have nucleus, and these cells, therefore, are called prokaryotic. Now, multicellular organisms are organized in a hierarchical manner. You have already studied the emergent property of life. When things organize together in a special way, the combined form produces different characteristics. Cells are the basic structural unit. Similar cells are organized and perform one kind of task, and these cells that are interconnected for a common function are called tissues. Different types of tissues are connected to form an organ. For example, a heart, a kidney, lungs, etc. These are organs. Several organs are linked together or connected together to form an organ system, such as the circulatory system, the digestive system, the respiratory system. Now, all these organ systems then combine or work together to form an organism. You and I have multiple organ systems working together in order to make a functional organism. Most cells are very small and you cannot see them by the naked eye. Microscopes are instruments that make small things appear big, a process called magnification. This is an example of a light microscope. This is an example of electron microscope, transmission electron microscope. Light microscopes can magnify an object up to 1,000 times, but electron microscope can magnify a specimen over 1,000,000-fold. Two things are important in microscopy. They are magnification and resolving power. Magnification is the process of enlarging an object. The object looks much bigger compared to its real size. This is an example of magnification. So, if you have one-to-one magnification, 5 times magnification, you can see more detail when you magnify 5 times, but you see less of it. You see more and more of less and less. The second parameter that is important in microscopy is a thing called resolution. Resolving power is the ability of a microscope to distinguish two adjacent structures as separate dots. In this case, it is difficult to distinguish one dot as separate from the other. So this is looking blurry, whereas the same organism under electron microscope has a much clearer, crisp image because it has a greater resolving power. Microscopes with different optical systems produce different images, and you can use even light microscope for a variety of different applications. So, for example, you may use phase contrast microscopy to visualize cells that appear like three-dimensional. Here you can see even clear cells to be having some structure. In this kind of microscopy, the optics or the glasses exploit the different density of these internal structure and then create a contrast. You may also use chemicals called stains, and as a result, you will see better contrast. Electron microscopes use electron beams rather than light. Electron beams have a very short wavelength, and as a result, they have a much higher resolving power. We typically use two kinds of electron microscopy. One kind slices the tissue or the specimen into very, very thin slices and then expose these to those electron beams. This is called transmission electron microscopy. The electron beams go right through that thin section of the specimen. And as a result, areas that are really dense, they are not permeable to electrons. They appear dark in color, whereas specimens that let electron beams go through appear white in color. So these are reasons where there is not a lot of electron permeability. Here, there's a lot of electron permeability. Here, very little electron permeability. The other kind of microscopy is called the scanning electron microscopy. These are red blood cells, and the surfaces of those red blood cells are scanned. For this technique, they use microscopic gold particles to coat the cells completely. So the exterior of the material that you are going to view will be coated with thin gold particles, and then you pass the electron beams. Those electron beams bounce back from the gold, and as a result, you can create an image. This is really good to visualize the surface features, the contours, whereas the transmission electron micrograph is better suited to study the internal details. Over many years, microscopy has advanced rapidly. Microscopes were first invented back in 1595, early 1600, and people have been fascinated with microscopic organisms. They view all kinds of organisms that were previously unseen. New microscopic world became accessible to human beings after microscopes were invented. So by 1830, scientists began to realize that all plant cells and all animal cells, in fact, all organisms are made up of at least one cell. And therefore, the idea of cell theory emerged. Cell theory is a basic principle of biology, which really states three things: that cells are basic units of life. What does that mean, basic unit? It is the basic structural unit, meaning there is a minimum structure, and basic functional unit, which is the life's function. So think about this one. What is the minimum structure required for something to be alive, for a sack of fluid chemicals to be alive? That is the structural part. And a cell inside its cell membrane boundary has the right chemical constituents that makes it alive, that gives it life. The functional unit part, function includes life's function. So that means, what does a cell do? For example, pass its genetic information to the next generation, meaning reproducing, meaning digestion, utilizing energy, being sensitive to the environment, and so on and so forth. So these are the basic units of life. Second idea is all organisms are made up of at least one cell. You already saw some organisms have many cells, others have just one cell, one organism. Lastly, the idea that cells come from pre-existing cells, meaning that cells do reproduce. So please pay attention to these three principles of cell theory. You will be asked about cell theory in the exam. This is an important concept for you to recognize. Now let us delve into the cell itself. There are four common cell components. This is the structural part. First, you have a plasma membrane or cell membrane. In this diagram, the cartoon, you see a cell wall, which is the outer covering, and right below the cell wall, there is a membrane called the plasma membrane or cell membrane. The cell membrane is the boundary that separates living things inside the cell and non-living part on the outside. The fluid that is inside the cell membrane is called the cytosol. The space between the nucleus here and the membrane here, that is called the cytoplasm. So in the cytoplasm, the fluid part is embedded. So oftentimes students confuse about the cytosol and cytoplasm. If you had a very microscopic filter paper and you could strain all the fluid part out of this area, that would be the equivalent of the cytosol. Now, cytoplasm includes organelles, everything between the cell membrane and the nucleus. Within the nucleus, there is DNA. DNA is the genetic material of the cell. And these tiny dots here, black dots that are in the cytoplasm, they are called ribosomes. Ribosomes are factories where proteins are synthesized. Now, broadly speaking, cells fall into two categories: prokaryotic cells and eukaryotic cells. Prokaryotes lack membrane-enclosed internal compartments such as the nucleus. And they have a cell wall which is different from other cell walls such as plant cell wall or the cell wall of fungus. Bacterial cell walls, a prokaryotic cell wall, contain a chemical called peptidoglycan. Prokaryotes are believed to be much like the first or primitive cells. That's why they're called prokaryote. Karyon means nucleus. Organisms in the domain archaea and bacteria are prokaryotic. Now, this is a generalized structure of a bacteria, which is a prokaryotic cell. This one includes a capsule, cell wall containing peptidoglycan. Inside the cell wall, you have the cell membrane. And within the cell membrane, you have the cytoplasm, and the DNA is floating around in the cytoplasm. This region where DNA is found is called nucleoid region. Prokaryotic cells are much, much smaller than eukaryotic cells. These cells have a much larger surface area compared to their volume, and therefore they are very efficient in diffusion and movement of chemicals compared to the eukaryotic cells. Typical cells are very small, whether they are prokaryotic cells or eukaryotic cells. Why are they so small? It really has to do with simple physics. If you make a thing really small, it has a larger surface area compared to its volume. As you increase the size of the object, any object, the area increases slowly compared to the volume. Volume increases rapidly because volume increases by cube compared to the area which increases by square. Having a large volume and a small surface area slows down diffusion, movement of substances inside. In this case, you have a larger surface area compared to the volume. Therefore, movement of molecules inside, mixing of it, moving things in and out becomes much easier. This is why cells are smaller, because they need to be efficient in transporting substances in and out. This is an illustration of a typical eukaryotic cell. Eukaryotic cell has many, many parts. You will be given additional pictures like these, and your really main task is to understand, to really memorize the names of all these different parts and their function. What does each part do, and what is its function? So that is the part that you really need to understand. Okay, and we will cover one at a time. So for example, there is a structure called flagella. This is a centrosome. Centrosome is important during cell division. This one will produce the structures called spindle fibers. Inside the cell, we have these small thread-like structures called cytoskeletons. Cytoskeletons have three major kinds of fibers. These are called microfilaments is one kind, intermediate filament is another kind, and microtubules are a third kind. You have a structures called mitochondria. Mitochondrion is singular, mitochondria are plural. So these mitochondria are organs where food is burned and energy is released. So mitochondria provide cells with ATP, energy for Lysosomes are structures that will digest food, break it down, perform hydrolysis. Peroxisomes are structures that will that are like it's like its power plant. They will incinerate, they will burn, and they will dispose some of the toxic substances that are a byproduct of many biochemical reactions. Now, Golgi is a structure that will process the proteins and target it to different structures. This is almost like FedEx or US Post Office that takes the substance from endoplasmic reticulum, modifies it, and ships it to different destinations, including secretion, which is to the outside. This one is nucleus here. Nucleus contains chromosomes and chromatins inside the nucleus. This structure is called nucleolus. Nucleolus are the site where ribosomes are synthesized. You see in the cytosol, there are many, many ribosomes. These ribosomes are sites where proteins are made. Some of these ribosomes, once they begin making protein, they attach to the endoplasmic reticulum, and these are called rough endoplasmic reticulum. You have structures like this that have no ribosomes attached to them. These are smooth ER, smooth endoplasmic reticulum. So endoplasmic reticulum themselves are these internal membrane-bound structures that will make proteins and house proteins, and those proteins then move from ER, endoplasmic reticulum, to the Golgi. Now let us consider these cell parts one at a time in detail. First, eukaryotic plasma membrane. Plasma membranes are made up of phospholipid bilayer. This type of organization is called a fluid mosaic model. The fluid part is the lipid part here, the phospholipids that form the bilayer. The mosaic part is all these different kinds of proteins that are embedded or are floating in the phospholipid bilayer. So they constitute the mosaic part. What are such proteins that are in the membrane? So they are channel proteins here, for example. This is a channel. It has got a hollow structure. So this channel will allow movement of molecules from outside to the inside. You may have proteins such as this channel that span the entire bilayer. These proteins are called integral proteins. Sometimes we have proteins that are present only on one side, one half of the bilayer, not on the other half. It just does not span all the way through. Now, these are peripheral proteins. So we have integral proteins, we got peripheral proteins, we got channels, and we got enzymes, and these proteins can be modified too. So for example, you have this is a sugar chain. Sugar chain is attached to lipid here, so phospholipid, so this is called glycolipid. You have a glycoprotein here, so protein sugar chain is attached to this glycoprotein. You also have these structures called cholesterol. Cholesterol are found in animal cells. These cholesterols will help make the cell membrane a little stiff, more dense, and it'll maintain the fluidity of the membrane. Cell membrane becomes less fluid when it is too hot, or more fluid when it is too cold. So this is really helpful in buffering the integrity of the membrane. Now, in the cytoplasm, many of these membrane proteins or lipids are interconnected with the cytoskeletal fibers. So these are filaments of the cytoskeleton. And we will talk about what those cytoskeletal filaments are in a moment. Now, the cytoplasm is next. It is the region between plasma membrane and the nuclear envelope. This consists of organelles that are suspended in gel-like cytosol. Cytosol is the fluid part, the liquid part, and the cytoskeleton. Now, 70 to 80% of the cytoplasm is water, but because of the sugars and all other substances dissolved in it, it has got this semi-solid, viscous consistency. Now let us take a look at the nucleus. Nucleus usually is the largest organelle in the cell. This is the organelle where genetic material, DNA, is housed. If you go from outside of the nucleus to the inside, these are the things that you may encounter. First, nucleus is connected to the endoplasmic reticulum. There is a continuous membrane stacks that extend from the outer nuclear membrane to the endoplasmic reticulum. It is continuous. It is one with the endoplasmic reticulum, which is embedded in the cytoplasm or cytosol. Nuclear membrane itself is a double membrane. What does that mean? It is a two bilayer, the outer bilayer, and there is an inner bilayer. The outer bilayer, as I said, is continuous with the endoplasmic reticulum. Now, inside the nucleus, there is a fluid, and that fluid is called the nucleoplasm. The dark staining structure, very dense structure found inside the nucleus is called nucleolus. Nucleolus is the site where ribosomes are made. Nuclear envelope separates the processes called transcription and translation. Recall, transcription is a process that makes RNA from DNA. This process happens inside the nucleus. Translation is a process that manufactures proteins. Now, those proteins are manufactured in the ribosome. Ribosomes are found in the cytosol, or they are embedded in the endoplasmic reticulum. There are many nuclear pores that are organized that perforate the nuclear membrane. These pores, nuclear pores, connect the nucleoplasm to cytoplasm. They do regulate the flow of molecules back and forth, and there are special kind of transporters, helpers that are required for nuclear localization. So if a protein has a nuclear localization signal, this is going to pass through the nuclear pore and go on the inside and stay there. Now, nucleolus is the region inside the nucleus where ribosomes are assembled from RNA and proteins. Ribosomes have two subunits, large subunit and a small subunit. Here is a structure of a ribosome. In the process called translation, ribosomes have two subunits. This is the large subunit. Here is a small subunit of ribosomes. The large unit and small subunits are separately manufactured in the nucleolus region, and they are independently transported to the cytosol. And in the cytosol, when these ribosomes encounter messenger RNA, they assemble in this format. And ribosomes provide a platform on which the code of the messenger RNA can be read to manufacture proteins. So these are polypeptides, amino acid chains. Each amino acid is brought to this manufacturing site by transfer RNA. So this is an example of a transfer RNA. Transfer RNA matches to the messenger RNA, and each transfer RNA brings a unique amino acid to the protein production site, and a large growing polypeptide chain is assembled like that. Mitochondrion is another important organelle. This is the site for conversion of stored chemical energy to useful chemical compound called ATP. Cellular respiration deals with how ATP is produced in the cells. The mitochondria itself has an outer membrane followed by an inner membrane which is highly folded. These folds are called cristae. The cytosol or cytoplasm of mitochondria, this portion here indicated blue, this is called the matrix. This is like the cytoplasm of mitochondria, matrix. and the space between the outer membrane and the inner membrane, this is called the intermembrane space. Peroxisomes are membrane-bound organelles. They are found in virtually all kinds of eukaryotic cells. Peroxisomes are oxidative organelles, and in peroxisomes, molecular oxygen serves as a co-substrate and produces hydrogen peroxide. The name peroxisome comes from the production of hydrogen peroxide, which can be pretty toxic. In peroxisomes, lipids and amino acids, fatty acids are broken down. Part of that byproduct involves production of hydrogen peroxide. This organelle is responsible for detoxification of different kinds of poisons, including hydrogen peroxide. Although both plant and animal cells are eukaryotic, there is a difference in the structure of plant cells and animal cells. Some of the differences include the appearance of cell wall. Plant cells have a very strong cell wall. Animal cells lack cell wall. They simply have cell membrane. Plant cells, of course, also have cell membrane. Additionally, animal cells have a structure called centrosome. Centrosome is an organized bundle of microtubules. Both plant and animal cells have microtubule organizing centers called MTOCs. But animal cells have centrioles associated with the microtubule organizing centers. Plant cells do not have lysosomes, but animal cells contain lysosomes. Lysosomes are equivalent of cell stomach. They digest the food ingested by animal cells. Although both plant and animal cells are eukaryotic, there is a difference in the structure of plant and animal cells. Both cells organize microtubules into spindle fibers. Microtubules in plant cells originate from what are known as microtubule organizing centers. These centers are called centrioles in animal cells. Two centrioles come together to form a structure known as centrosome. Centrosome plays a key role during cell division when it splits and moves to the opposite poles, and it begins to generate the spindle fibers. Plant cells and animal cells also differ with respect to the presence of lysosomes. Lysosomes are digestive organelles of the cell, similar to our stomach. In plant cells, you do not see lysosomes, but because animal cells ingest food, they need to perform that digestion, which happens in the lysosomes. Lysosomes are also acidic, just like your stomach is acidic in nature. They have low pH. Finally, plant cells have cell walls, chloroplast, and other specialized plastids, such as leucoplasts. They also have a central vacuole. Animal cells do not have these structures. Now, here is another image of an animal cell. Notice you have a cell membrane here, and different internal organelles, membrane-bound organelles, are shown here. That includes mitochondria. Here is a Golgi body or Golgi apparatus, vacuoles. You have lysosomes, peroxisomes, and there are vesicles. Vesicles are these small packets, bubbles of phospholipid-enclosed organelles. And those vesicles blend with the cell membrane and release the content to the outside. These are called secretory vesicles. These small dots that you see are ribosomes. Ribosomes are sites where proteins are produced. You have this endoplasmic reticulum here. In this case, these red dots lining the endoplasmic reticulum are ribosomes. Because of the presence of ribosomes, such endoplasmic reticulum, is known as rough endoplasmic reticulum, rough ER. You have smooth ER here. Smooth ER is responsible for synthesis of lipids and hormones. These smooth ERs lack ribosomes. You have the nucleus, nucleolus, and chromatins. Chromatins are DNA threads that are wrapped around histone proteins. Here is an example of plant cell. Now, please study these diagrams carefully and note a few similarities and differences. So this is centrosome. They consist of two centrioles. Each centriole contains a bundle of triplet microtubules. These are microtubules. We're going to talk more about microtubules later. These are triplet microtubules, and there are 9 such triplets organized. This is one centriole. There is a central single microtubule at the core too. These two centrioles are perpendicular to each other, and then they are, each triplet is separated by non-tubulin proteins, which is indicated in the green line, and these things hold microtubule triplets together. Now, plant cell walls are made up of cellulose. Cellulose is a long polymer of glucose. Here, the orientation of glucose molecules is flipped. So, this is alpha glucose, beta glucose, alpha glucose, beta glucose. So, the beta 1,4 glycosidic bond causes fiber-like appearance. So, these are fibers. Now, plant cell walls differ from bacterial cell walls because they contain the cellulose, whereas bacteria contain a structure called peptidoglycan. In the lab, you will see onion cells here like that. These are onion cells, plant cells, and you can see very strong cell wall and the nucleus and cytoplasm. Chloroplasts are the green organelles that are responsible for photosynthesis. Chloroplasts contain two membranes. This is the outer membrane of the chloroplast. There is an inner membrane of the chloroplast, and inside the inner membrane, there is a stack of thylakoid membrane. These thylakoid membranes are like flattened fire hose. So they themselves are tubes, and a lot of green pigment called chlorophyll and various light harvesting proteins are embedded in the thylakoid membrane. Photosynthesis involves two sets of biochemical reactions. In the first step, light is harvested. So the whole light harvesting complex is located in this thylakoid membrane. ATP is also synthesized during the light reaction. Now, that ATP and a reducing compound called NADPH are produced in light reaction. They then are used to make sugars in the stroma of the chloroplast. Stroma is a structure similar to the cytosol of the cell. Stroma is the fluid that is inside the inner membrane of the chloroplast. Plant cells have large cell vacuole. This large cell vacuole is also known as central vacuole, and it helps regulate water concentration under different environmental conditions. If the plant cell is exposed to a hypertonic solution, meaning salty solution, water from inside the cell leaves, and the cell undergoes a process called plasmolysis. In an isotonic solution, there is equal movement of water in and out. Equal amount of water can go out or in. So they are in equilibrium. In hypotonic solution, this is a solution that has very little salt compared to the inside of the cell, compared to the cytoplasm of the cell. Because the cell cytoplasm is hypertonic, outside is hypotonic, water begins to move inside and the cell appear very turgid, very bloated. Both mitochondria and chloroplasts developed in the cells as a result of a process called endosymbiosis. These organelles are like bacteria because both chloroplast and mitochondria also have DNA. They have protein-making apparatus, including the ribosomes. And the nature of the DNA and the ribosome and the way they manufacture the proteins are very similar to the prokaryotes. Lynn Margulis hypothesized that mitochondria and chloroplast originated as independent prokaryotic organisms. The prokaryotic ancestors of the eukaryotes ate these guys up. Once they were swallowed, instead of getting digested and utilized for the macromolecules, the chloroplast and mitochondria began to perform useful function for the host cell. And then over time, they have now become permanent resident of the cell. Without mitochondria, neither plant nor animal cells can survive, and plant cells without chloroplast are not able to perform photosynthesis. Now let us take a look at endomembrane system. Endomembrane means internal membrane. It consists of internal membranes that are found embedded in the cytosol. There is a network of these membranes, and these membranes can jump from one organelle to the other organelles. All of these membranes are made up of phospholipids. These internal membrane structures are found in nuclear envelope, lysosomes, vesicles, endoplasmic reticulum, Golgi apparatus, and the plasma membrane. Endomembranes can move from one organelle to the other organelle in the form of vesicles. So there is an elaborate vesicular transport system within the cell. Lysosomes are like the stomach of cells. These organelles break down food and large macromolecules, including some worn out organelles. Say our mitochondria is too old and unable to perform its function, or a cell is dead. So all that debris is cleaned up by the lysosome. Lysosome contain inside the digestive juice, digestive enzymes. They have acidic pH, and they break down macromolecules into their respective monomers, which will be used as a raw material, as a starting material for biosynthesis of other macromolecules. The most extensive component of the endomembrane system is the endoplasmic reticulum. These endoplasmic reticulums are interconnected sacs of tubes. And in the ER, ER can be smooth ER or rough ER. Smooth ER lack ribosomes, whereas rough ER contain ribosome. Inside of this ER, endoplasmic reticulum, the hollow portion, the pipe inside the tube is called the lumen or cisternal space. Newly synthesized proteins are inserted into the lumen, and a part of that ER pinches off and comes out as a vesicle and is then transported to another structure called Golgi apparatus, where this protein undergoes further modification. Now, endoplasmic reticulum, as I mentioned, is rough or smooth, okay? Rough ER makes phospholipids for cellular membranes. Phospholipids or modified proteins are not destined to stay in the rough ER. They reach their destinations via transport vesicles that bud from the rough ER's membrane. Okay. Smooth ER, this is an example of smooth ER. They lack ribosomes, and therefore they do not perform protein synthesis. These structures are involved in metabolism of lipids. These are some of the functions of smooth endoplasmic reticulum. They synthesize carbohydrates, lipids, steroid hormones, and they are also responsible for detoxification of medications and poisons. You have a lot of smooth ER in the liver cells. ER also are a warehouse storage for calcium. That calcium is essential for movement of our muscles. This is a muscle cell. Cell membrane of those muscle cells are called sarcolemma. Sarco is a muscle. So they have endoplasmic reticulum of the muscle cells, which is called the sarcoplasmic reticulum. Sarcoplasmic reticulum has this terminal cisternae, T-tubes, etc. So in the lumen, they have a warehouse of calcium. When electrical impulse is generated, those calciums are released through these T-tubules into the muscle cells, which causes the contraction of the muscle. Now, Golgi apparatus. Golgi is a structure. This is like a shipping yard, like a processing plant. In Golgi, lipids or proteins inside the transport vesicles, they need to be sorted, they need to be processed, and they need to be targeted to different cellular destination. Some of these need to be secreted. So from ER, the vesicles move to the Golgi and they dock on a site called the cis side of the Golgi. And these content, these proteins or lipids, they are then processed and they gradually move from one cisternate to the next one, to the next one. And finally, from the other end, the trans end, fully processed vesicles emerge. And these vesicles have their own target destination. Some of them are targeted to the nucleus, others are secretory proteins, others stay within the cytosol as vesicle. Often students have this misconception about the cytoplasm being amorphous, without shape, and like viscous liquid. That is far from truth. Cytoskeleton is really cluttered with a lot of protein fibers. And these fibers collectively are called cytoskeleton. Cytoskeleton helps maintain shape of the cell, hold different organelles in their respective positions, allows the movement of the cytoplasm within the cell, and also enables cells within multicellular organisms to move. Amoeba, for example, or our phagocytes in our bloodstream, they can crawl with the help of the cytoskeleton. Now, three components of cytoskeleton, three kinds of cytoskeleton fibers include microfilaments, intermediate filaments, and microtubules. Microtubules are large, microfilaments are small, and intermediate filaments are in between. So for example, microtubules, roughly they have 25 nanometer in diameter. Intermediate filaments have 10 nanometer in diameter. Microfilaments are 4 to 5 nanometer in diameter, 4 to 5 nanometer in diameter. So this cell here, this cartoon shows different kinds of protein fibers. You have the microfilaments or the actin filaments. Okay, you got the intermediate filaments here, and you got the microtubules. So now take a look at the microfilaments. Microfilaments are made up of actin polymers. This is an individual globular protein called actin. Now, actin protein linked with other actin to form a long actin polymer, and two such strands are intertwined to form the actin filament, to form the functional active filament. Microtubules are like a hollow pipe. Imagine you have a straw, so they look like that. They are made up of tubulin proteins. There are two kinds of tubulin proteins, alpha tubulin and beta tubulin. Now, these microtubules serve as a track, as a train track for vesicles. So vesicles move along these tracks, and there are some motor proteins such as dynein, kinesins, myosin, etc., that move along these microtubule tracks. Now, cilia and flagella are structures that are found on the outside of the cell. Our respiratory tract, for example, reproductive tracts, they are lined with cilia and flagella. Both cilia and flagella have similar structure. It's just the flagella are very long and a few, but cilia, there are many, and they are short. Both of them have a structure known as 9+2 structure. So there are 9 doublet microtubule pipes. So these are 9, one doublet, two doublets. If you count them, there are 9 microtubule doublets. So that is 18 microtubules. And there is a doublet on the inside, okay? Now, the whole structure is then surrounded by a cell membrane and a cytoplasm. So this is the structure of cilia. This is also the structure of flagella. This is called 9+2 structure because there are 9 doublet pipes plus 2 here at the center. So 9+2 arrangement. As you see, cells are highly organized, but the organization goes beyond the internal structure of the cell. Cell's exterior is also highly organized. There is almost as much story about the extracellular structure as it is of internal cellular organelles. For plant cells, outside of the cell membrane, there is a strong cell wall. This cell wall provides support. It's a barrier for infection. Plant cell walls are studded with small holes called plasmodesmata, and these plasmodesmata help connect one cell to the adjacent cell. In animal cells, they have different kinds of proteins on the extracellular area. This is called the extracellular matrix. Three major components of the extracellular matrix in animals include collagens and other fibrous protein, glycoproteins, these are proteins with sugars, and proteoglycans, protein in sugar chains. And they also have different linking proteins. Now let's take a look at this extracellular structure. So take a look at this extracellular matrix of animal cell here. So you have the phospholipid bilayer, you have these integral proteins, here is an integrin. So these proteins are connected, integral proteins are connected on the outside with fibronectin to collagen fiber. These are collagen fibers, okay? Now on the inside, these integrins are then connected to the microtubules. So these are microtubules. All the cells are interconnected in a tissue. You know, there is an adjacent cell on the other side that would be connected to this extracellular matrix. These green things, you know, these are called proteoglycan complex. Now the proteoglycan complex has a long polysaccharide to which a protein chain is attached. Okay, so this is called the proteoglycan structure. There is a core protein, and then there are these carbohydrates also sticking out. The figure below is an in situ fluorescence microscopy on C. elegans. C. elegans is an experimental model organism, a tiny nematode that has got set number of cells. Here the green color is resulting from green fluorescent protein that indicates extracellular matrix. And you see how the cells are lined up on one side of the extracellular matrix versus the other side. So these cells are interconnected. This type of model is used by scientists to study cancer. So for example, this cell that is colored purple is migrating to the other side. So cancer cells migrate from the area of initial tumor to the malignant tumor site at a different site. For that, they need to cross this basement membrane, which is made up of extracellular matrix. In this case, this particular cell is defective and is unable to go from this side across to the other side of the extracellular matrix. Now, cells also are interconnected by different kinds of cellular junctions. These intercellular junctions provide direct channels for communication between cells, and that happens differently in plants and animal cells. So for example, in plants, you have structures called plasmodesmata. Plasmodesmata are tiny holes on the cell wall. Plasmodesmata are channels that pass between cell walls in plants, and they connect cytoplasm to allow materials to move from cell to cell, especially small ions and water molecules can move from one cell to the next cell. Now, tight junctions are present in animal cells. These are watertight seals that prevent materials from leaking between the cells. They are found in the gut epithelium, our epithelial cells. are connected, so our gut is not permeable to water. So these are like rivets here. So these two plasma membranes, and they are connected by tight junctions. Nothing can go through these tight junctions. If you look at this diagram here, our intestinal cells have a polarity. So this side here on top is the lumen side, which is the inside of our intestine. Food is passing through here. Molecules need to be absorbed. Those molecules cannot enter through these sides. They need to go through this structure. They need to go through the membrane. So between these two cells, you cannot have food material going through the gaps between the two cells. So they do have very tight junctions, which forces the food to simply pass through the apical surface of the epithelial cell. Now, desmosomes are made up of intermediate filaments. These desmosomes these are short proteins in the plasma membrane. They are made up, these proteins are called cadherin proteins. And these proteins act as spot welds. So basically, these are cadherin proteins. Here's a desmosome on this side, desmosome on the other side. And these proteins hold the two cells together like rivets. They join the adjacent cells in stretchable tissues such as heart, lungs, muscles, etc. These are only found in animal cells. Now, gap junctions connect animal cells. These are gap junctions. There are many gap junctions between the two cells. This is one cell membrane of one cell. This is the cell membrane of the second cell. So you have these connection proteins, and there are 6 of these individual monomers that are organized into this structure. This is a closed conformation. This is an open conformation. Small ions, water, and other molecules can move through these open position from one phospholipid bilayer down this gap junction channel to the other cell membrane. Gap junctions resemble plasmodesmata in plants because they form channels that allow ions, nutrients, and other materials to move between cells. They are made up of 6 proteins called connexins, and they form a structure, elongated donut-like structure called connection here. So you have one connection, and the same thing is present on the other side. When connections from adjacent cells line up and they are aligned, gap junction channel forms between the connection. So this is a protein-lined pore that allows water and small molecules to pass between adjacent animal cells.